Hey guys, CRISPR here. Today we have a few small stories, but one really big one at the end of the video. So for our first small story, scientists at UC Berkeley have discovered how the proteins associated with CRISPR find their target. First, Cas1 and Cas2 identify the site in the genome where they insert viral DNA, which they can recognize later and mount an attack. Then, a third protein, IHF, binds near the insertion site and bends the DNA into a U shape, which allows Cas1 and Cas2 to bind both parts of the DNA at the same time. The authors of the study were grad student Addison Wright and postdoc Junji Liu. For our next story, Selectis has won the patent for the use of CRISPR in T cells in Europe. Selectis is a French biopharmaceutical company that specializes in T cell use for cancer immunotherapy. Hopefully they can use CRISPR to improve their treatment of cancer. For our next story, DARPA, or Defense Advanced Research Projects Agency, plans to invest $65 million in its Safe Genes project. Dr. Renee Wegerzin, the Safe Genes program manager, explained the purpose of the project. The field of gene editing has been advancing at an astounding pace, opening the door to previously impossible genetic solutions, but without much emphasis on how to mitigate potential downsides. DARPA launched Safe Genes Project to begin to refine those capabilities by emphasizing safety first for the full range of potential applications, enabling responsible science to proceed by providing tools to prevent and mitigate misuse. There are 7 teams for this project. All of the teams are led by some of the top scientists in the field. The first team is from the Broad Institute, led by Amit Chaudhry. The second is from Harvard Medical School, led by George Church. The third is from the Massachusetts General Hospital, led by Keith Jung. The fourth is from MIT and is led by Kevin Esvelt. The fifth is from North Carolina State University and is led by John Godwin. The sixth is from UC Berkeley and is led by one of the creators of CRISPR, Jennifer Doudna. The seventh and final team is from UC Riverside and is led by Omar Akbari. For our final story, and the big one that I told you about earlier in the video, scientists in the US have edited the DNA of viable human embryos. This was led by Shukrat Militapov of Oregon Health and Science University. It involved editing a large number of viable embryos and effectively correcting disease-causing genes. The embryos were developed for only a few days, and they were not implanted, so they cannot grow into babies. Militapov, who was in charge of this study, is very controversial. He was the first one to clone embryonic stem cells in humans, and he also invented 3-parent in vitro fertilization. In fact, he started working in China because he was not funded in the US. However, this gene editing was legal in Oregon, as long as there was no federal funding. This is huge news, and more editing of embryos are almost a guarantee for the future. They might even be grown into humans. One thing is for sure, Militapov is walking a fine line between science experiments and creating monsters. Anyway, that's all for today. This is CRISPR Man, signing off.